I'll be honest that I'm not surprised that it's you guys that are on here. Give it a little bit to hopefully people will get on here. Come on. Stop licking me. What do you want? Shh. Ripley, stop it. Mm-mm. Mm-mm. Hang on. Life was so hard as a dog. I don't want to wait too long because you guys are already here. I know that Eric is trying to get on. He's usually pretty good about joining these things, but well, just gonna have to wait till I'm done with this thing. All right, well, let's go ahead and get started. Hopefully, if other people join, they don't have a problem joining. Yeah, I should do this right here. Hey, be quiet. Seriously. She barks at anything. You put her out there with the person and she'll run away like, her life's depending on it. Stop. Okay, sorry, I'm trying to find a link for Eric. Here we go. No, no. I promise we're gonna start soon. I just. These slides. Oh, copy. This is it. Okay. All right, let's do it. Let's see, because of this. All right, here we go. Some microorganisms like influenza cause illness. What is the term for disease causing microorganisms?
Yes.
All right, good job, guys. So that's going to be pathogens, starting off with a pretty easy one. If you don't know that by now, then we're in a rough spot. All right, water treatment. It involves screening for serious pathogens by tracking populations of these. What's the term? Yep, so that's going to be coliforms. We call them, fecal coliforms typically, and that's what they'll track. And if there are coliforms in the water, then there could be potential for pathogens to be there, like Shigella. Okay, when performing tests to determine a pathogen's identity, we sometimes use differential media. What exactly is differential media? Okay, so differential media is media that will change color somehow, usually in response to some sort of metabolic byproduct in the whole process of breaking something down, like what we see with the urease and that sort of stuff. It changes color if it doesn't necessarily inhibit growth of one organism or another, then it's only going to be differential. You can tell the difference, but it doesn't inhibit growth. If it does inhibit growth, then the term we use is selective. Right. Atoms make up all matter in the universe. Which chemical bond involves transferring electrons from one atom to another atom? Reaching real far back. So this one's going to be ionic bond. Good on you guys for kind of splitting it up between covalent and ionic, because that's where we would go. Covalent will be sharing those electrons though. And if you have an actual transfer of electron from one atom to another, that's the ionic bond. Okay. pH affects biological molecules significantly. Increase in the pH means an increase in the hydrogen ion concentration. Is that true or false? All right, pretty fairly split here. So this one's going to be false. If you have a rise in pH, that would be trending towards the 14, as opposed to, remember, 7 is neutral. If we go below 7, like to 2, let's say, that's acid, and above is a base. So if you have a base, we're actually having less hydrogen ions, and acid has more hydrogen ions. And that is because, I'm not going to necessarily like go too crazy on this right now, but as far as the math was going with that, yeah, that's what I'm going to mention again. It was a negative logarithmic scale. So because it is the negative logarithmic scale, the lower the number, like 2 compared to 10, the lower the number, actually the higher the concentration of the hydrogen ions. So, right. Gotta love it. All right, some bacteria are motile, others are not. Are Fimbriae the structures bacteria use for motility? All right, so flagella are what bacteria use for motility. Fimbriae are for attachment. Okay, flagella are the structures used for bacterial motility, and they move via rotational motion. So do they move rotationally, or that's really what the question is going to ask here. So yeah, this one's true. So not only, like I said already in the previous question, flagella are the structures for bacterial motility, and they do move rotationally. So that flagella themselves in bacteria are actually going to be spinning via a rotor-like mechanism in the bacterial cell, which is true. For eukaryotes, they have flagella as well, but theirs move in a whip-like motion. So it is different. There is a distinction there. Okay, eukaryotes have membrane-bound organelles. Which organelle is the site where translation and protein modification occur? Okay, yep, that's going to be the endoplasmic reticulum. In the rough ER, which is studied with ribosomes, those ribosomes are what are going to kind of work with the messenger RNA to create protein during the process of translation. And then those translated protein products will go further into the endoplasmic reticulum to be modified however they need to be, if they need to be. So that's that process in the endoplasmic reticulum. A farmer steps on a nail in his farm. Skin represents which level of protection against infection? Yeah, it's the first line. Good job. This was originally like supposed to be a series of like linked together questions, but some of these just don't, I'm not testing you guys over anything related to it, so it doesn't link quite as well, but we'll get to it. All right, so the doctor that I guess the farmer went to begins by administering him a tetanus booster. What type of vaccine is a tetanus booster? Okay, so the tetanus vaccine is a toxoid vaccine. So it's actually a vaccine against the toxin of tetanus. And well, we'll get into it. I know that one of the questions coming up has to do with what I'm about to say, so I'm just going to leave it there. toxoid. It is vaccine against the toxin. So the tetanus toxoid has to be given in conjunction with other toxins. Why is this? Why do we have to do Tdap? Each of these are toxoids. Okay, so the toxoid vaccine that famously links together like Tdap or whatever version of it you get whenever you're whatever age or booster or whatever. Those guys are toxoids that are so small that our body cannot react to them without help from other molecules of some kind that we call carrier proteins. But when you're a tiny little thing that the immune system can't really react properly to on its own, you are called a hapten. So that is why we put them together so that they can help each other get proper reactions. Because they're too tiny otherwise. Right. What is a hapten? Oh, you guys, I have listened. Right, so this is going to be something that is too small to elicit a proper immune response. So forgot about that. So what type of immunity do vaccines confer? Okay, great. So yes, that is going to be artificial active. So it's artificial because it's made, we're giving it to you via medicine and you're not encountering it in like the wild or whatever natural environment of the pathogen. And active because we're eliciting a proper antibody response from your body and it will be protective for you as a result of that. So artificial active. All right, this one's a true or false. The purpose of the booster vaccine is to induce production of protective IgG by memory plasma cells. Yeah, so this one's true. Great job. Great. That's the whole point of kind of all of this, right? Develop that memory. In the primary immune response, which class of antibody is made first? Okay, the first time that you encounter something, your newly activated plasma cells are going to crank out IgM antibody first. Eventually, it will class switch to become IgG, which is the more specific one. IgE is going to be associated with like allergies, and then IgA is the mucosa, the secretory IgA involved in the mucosal membrane. Okay. All right, you can select more than one here. So what purpose does antibody serve? Okay, so it's going to be all of them. It's like my favorite thing with this one because there were 6 major ways that antibody could work for our bodies. This is 4 of them, basically. But yeah, it can also be an antitoxin. But anyways, let's move on. A lot of them are pretty much the same thing. They're just worded differently. Okay, we really only vaccinate against illnesses that cause harm. So what kind of harm does tetanospasmin cause? Right. So with tetanus, we're having the issue of the muscles cannot relax. So they seize up. It's called spastic paralysis. Those are fair bets either way. At least you guys are leaning the right way there. Clostridium tetani are gram-positive, endospore-forming anaerobes. What are anaerobes? Right. Yes, that's exactly right. Anything, they're organisms that require anything except oxygen as their final electron acceptor when creating ATP. Exactly. Good job. Okay. In aerobic respiration, what byproduct is made when oxygen works as the final electron acceptor? Excuse me. Yes, this is going to be H2O because we're taking the protons from that proton motive force. So we've pumped all those hydrogens, the H pluses that don't have any electrons, pump them all out. They come through ATP synthase to drive production of ATP. And then they're just hanging out there. We also pushed a whole bunch of electrons through all of those kind of electron transport chain cytochromes. So we got electrons hanging out. We've got hydrogens without electrons. So we bring in oxygen so that we can combine all that together to make H2O to take care of those waste products essentially. So we can kind of reset in a way and keep cranking out ATP. So yeah, water. We do make carbon dioxide obviously in the whole process, but that isn't necessarily linked to aerobic respiration. It's something that will occur in the Krebs cycle as well as right before the Krebs cycle. Which of the following is an enzyme that helps aerobic organisms deal with toxic oxygen products? See if you remember the name of that. Which one was the enzyme that did that? Yep. So this is going to be catalase, and it usually is divided between people's minds. Catalase or oxidase. Oxidase was referring to cytochrome C oxidase, which was involved in the electron transport chain. But when we're talking about dealing with those toxic oxygen byproducts like hydrogen peroxide, catalase takes hydrogen peroxide and turns it into water and oxygen, just like what we learned in the lab. All right, the tuberculin PPD skin test is helped to diagnose tuberculosis. What type of reaction is observed in this test? Remember. Okay, so the PPD skin test is type 4 hypersensitivity. Type 4 hypersensitivity has to deal with the CD8 and mostly just let's say the T cell response essentially. So they consider it to be cell mediated immunity. And if you think about it this way, when we talk about tuberculosis, and the bacteria actually lives inside of those alveolar macrophages, right? So that's what the whole problem was, because they try to kill them with more alveolar macrophages come into the lung, but the bacteria just go and live in it and it just spreads within them. And then we have to wall it off and create these areas in the lung that you can see on the x-rays. Well, so they like it inside the cells. So our body, the way it responds in the immune response would say there's something inside of this cell. So we need to kill that target cell. So that is your CD8 cytotoxic T cell that takes care of that. That's your cell mediated immunity. So yeah, we're testing to see if you have that response already built against tuberculosis, because if you do, then that means that you have seen it in some form, whether it is the actual illness or the vaccine. So yeah, that's what that's about. All right, you can select more than one on this. What other tests would be used to confirm tuberculosis diagnosis? Kind of talked about this in the review, if you guys were there for it. Great, so this is going to be chest x-ray and the acid fast stain of sputum. Yeah, so that's pretty much what we'll say. Mycobacterium acid fast. So it's only one genus that you have to remember for acid fast. Acid fast, mycobacterium, acid fast, always. Bacillus, Clostridium, endospore, Bacillus, Clostridium, always. So those are the kind of the two groups to cling on to. All right, drug resistance is becoming increasingly common. What are some ways that bacteria become drug resistant? All right, so it's everything except for parenteral. You guys mostly got that one. Yep. So transformation, remember that's picking up it's what we talked about in the lab, right? Where we acted out. None of those, I said we, but I acted it out. You guys watched me do it with the little containers and the pipe cleaners and whatever. But yeah, taking up DNA from outside of the cell. So exogenous DNA is transformation. Conjugation is when two cells are linked together by the sex pilus and then transfer DNA through there. And transduction is transfer of DNA via phage. Good job. Okay, drug resistance often arises from mutation. When UUA becomes UUG, but it still codes for leucine, what type of mutation is it? All right, so this one's going to be called silent. We'll kind of talk about the other ones too, because I feel like it's fair to kind of bring up and refresh our memory about them. Remember that in translation, which is going from mRNA to protein, right? And as we are, got our little ribosome on the mRNA, and the transfer RNAs come in carrying amino acids. We match up the 3 nucleotides on the anticodon of the transfer to the codon 3 nucleotides of the mRNA. So, A to U and G to C, because we use U when it's mRNA or RNA. So that's kind of the process of what we're talking about here. It's always going to be sets of 3, and a set of 3 on the mRNA, the codon will code for an amino acid. Some amino acids, many actually, are coded for by more than one codon. So in this case, we have one codon, UUA, and it had a mutation happen that made it change into UUG, and it will still, in the translation process, code for leucine as the amino acid. So that's a silent mutation, because in the end product, the protein itself isn't going to be changing at all. All right, so let's go to nonsense. If we had changed the UUA into a codon that didn't code for an amino acid, that is also known as a stop codon, that causes the whole ribosome to dissociate and just stop, right? We're done. That is a nonsense mutation. You definitely don't want nonsense mutations popping up in the middle of your protein when you're not done. Nonsense, the way I remember that one is like, it's total nonsense, then not even the ribosome or the tRNA can read it. So that's going to be the stop. It's always stop if it can't make an amino acid. And then we have missense. Missense will be just change. So that's going to be change UUA. If UUG coded for a different amino acid, changing from leucine to, let's say, alanine or something, then that would be a missense. We've got a different amino acid than what should be there. Then we've got a problem called a frame shift mutation. If we delete an amino acid in the whole sequence, that would be like, or add a new, not an amino acid, sorry. If we delete a nucleotide, like it was UUA, instead of changing it to UUG, let's just delete the A and shift everything downstream up. Well, now suddenly what was codons that would have read, maybe UUA now is reading UUG, but the next steps that would have been GGG, now they're reading GGC because it all got shifted over by one. It is like taking out like a word or a letter, let's say in a book, and then shifting everything over, but keeping the size of the words the same. You wouldn't be able to read it. So that's what a frame shift mutation is. We keep the actual like 3 has to be that way all the time. But if you take out or add a nucleotide, then it's going to shift basically which codon, how you read the codons essentially. So yeah, so that's a huge problem. It changes everything downstream from whatever that mutation is. Frame shift and nonsense, man, they're the worst of them. All right, good job. Drug resistance is partly due to the prevalence of tuberculosis in the AIDS community. What type of hyposensitivity is AIDS? All right, so hyposensitivity for this one is going to be secondary. So primary is going to be genetic hyposensitivities, and then secondary is like acquired hyposensitivities. Yes, Lorenzo, I am going to post the recording of this on Canvas as well as the actual like Kahoot. I'm going to make an assignment so that you guys can go through it as much as you would like to. Sorry, I just saw that. I don't know how long ago you posted it. I guess I could check it, but.
Hooray.
All right. Okay, that same farmer returns with a black eschar near the puncture site of wherever I guess I'm touching the hand because it's always the same picture. Where he stepped on the nail, I guess. So is this a sign or is it a symptom? We already know what disease this is going to be. We've seen it in math class. This wasn't my number one example. Yes, this is going to be a sign. Great job, guys. Yeah, good job. Remember, symptoms are going to be subjective. So pain and things like that you can't measure. Signs you can. So what disease is associated with the black eschar? See if you guys remember our good old friend. Yep, most of you guys got this one. This is going to be cutaneous anthrax. The black sore is always indicative of that. You don't really see it much in any other disease. Not that way, so contained. So blood samples collected, and we order a CBC. What is the most numerous leukocyte in a typical adult? If you were to order a CBC. Let's see. Yep, neutrophil. Great job. So yeah, it's going to be the neutrophil. I don't know how else to talk about it other than that. Neutrophil. It's a granulocyte. The doctor confirms the anthrax diagnosis via PCR. So what are the main steps in PCR? Yeah, so most of us got this one. It's going to be denature, which is splitting apart the double strands, annealing the primers because we have to have something to build off of, and then elongating from there, adding on nucleotides via the polymerase, the DNA polymerase, which happens to be Taq polymerase is what we use because it is stable, heat stable. We should have heating and cooling. So you got to be stable. So anthrax is technically a zoonosis. What does zoonosis mean? So cute. Okay, so zoonosis just means that animals are going to be the natural reservoir. So primarily associated with animals. Let's see. So what type of medication would be used to treat anthrax? Okay, so pretty good here. It is a bacteria, so bacteria are going to be treated with antibiotics here. Bacillus anthracis is the name of it. So the doctor opts to treat with trimethoprim, which works by inhibiting folic acid synthesis. Is that true or false? Basically, how does trimethoprim work? All right, so this is in fact going to affect folic acid synthesis. It's like a sulfa category drug. All of the sulfas are going to be affecting the folic acid synthesis. Now, most of them have sulfa in the name. Trimethoprim is the only one that doesn't. But if it helps you remember, metho, like methane smells awful, sulfur smells awful. Just stick it in with that group. I don't know, it's silly, but that's how I kind of remember that it falls into that category. Okay, the farmer is going to recover from his anthrax, but will develop a painful scar, and that scar will linger for decades. What is the term to refer to this? Yeah, so the term for this is sequelae. Sequelae means anything that lasts, a lasting effect of a disease after you have cleared the disease. It can be much worse than that, right? So Bacillus anthracis produces hardy endospores. Endospores are readily destroyed by most surfactants. True or false? You're cleaning your area, maybe your patient's bed. It was this farmer. He was on his bed. He was rubbing that Eschar all over everything. Are surfactants going to be enough to kill it? That's right. So no, it won't be. It won't actually kill endospores. Surfactants are not sterilizing. That's just a matter of remembering that kind of chart or whatever that we set up of which were sterilizing and which weren't. So how would you kill endospores like those produced by Bacillus anthracis? Which the following would be effective? All right, so autoclave is the only one of these choices given that actually sterilizes, which means it will kill everything, including the endospores. Now we do remember, and I'll mention just one more time, prions aren't killed by traditional autoclave. You have to go much higher temperature, 135 instead of 121 degrees Celsius, right? And treating with sodium hydroxide before that to help denature the protein. It's a very strong base. Just thought I'd mention it. Oh my God. No wonder I think of these things. I guess I think of them because I know what's coming up. True or false? Traditional autoclaving is effective for inactivating prions. Yeah, I guess I guess I pay attention to my own freaking slides. All right, yeah, that's false. Yeah. Watch it go over all like the things you can do next. Okay, not really. So in activating prions, you have to denature. What level of structure is the alpha helix? So if we're getting denaturation, bring it to primary. What level is the alpha helix? So, and the beta sheet, but I can't fit both of them on there because it's words have limits on how many I can put. Right, so that's gonna be secondary. Great job. I know that's one that we've kind of gone over a bit routinely, so a lot of you guys are holding on to that one. It's great. So what type of disease do prions cause? All right, so it's a spongiform encephalitis. We consider it an acute encephalitis, and more specifically, we'd say spongiform encephalopathy because it's a little bit different from a true encephalitis, but yeah. All right, what type of genome do the prions have? Right, so they are not going to have a genome, they're just protein. Literally, that's all is a protein. So prion associated disease is always fatal. What types of microorganisms would decompose your body after death? Saprobes, that's exactly right. Things like mushrooms, and there'll be other organisms as well, bacteria and stuff that will break down, you know, decaying detritus and stuff like that. 11-year-old Erica presents with a cough, mild fever, and difficulty breathing. The doctor diagnoses pneumonia, which is not kind of what you were expecting when you saw the choices, huh? All right, so a little bit of I don't say a ruse going on here, yeah, but the answer is syndrome. That's the only one that really applies to all of it. As far as pneumonia goes, are we always going to treat pneumonia with antibiotics? There are viral and fungal pneumonias as well. Typically, it's going to be bacteria. In her case, it probably is going to be bacterial, but pneumonia itself is not always bacterial. So keep that in mind. Right. The doctor orders an indirect ELISA to test for what he thinks is the cause. What does indirect ELISA look for? Right. So it is going to be antibody. I don't know. That's just one you got to remember. After ELISA comes back, it is confirmed that she has pneumonia by the most common cause of pneumonia, which is great job, guys. Yes, that is going to be Streptococcus pneumoniae. Which of the following is another disease associated with Streptococcus pneumoniae infection? Excellent, guys. So yes, it is the most common cause of meningitis, in addition to being most common cause of pneumonia. It is the pneumococcus. There is a vaccine available. Streptococcus pneumoniae is only a pathogen and is not part of anyone's normal biota. We went over this one in the review, so it's probably a refresher there. So yes, that is going to be false. You absolutely can harbor it as part of your biota. We talked about this with mothers getting swabbed for group B streptococcus. About 10 to 40% of mothers will have it in their vagina, and this is one of those bacteria that qualify in that category. So the doctor treats Erica's pneumonia with amoxicillin. How does amoxicillin work? Okay, so that's right. Most of you guys got this one. It is going to be cell wall. Remember, there were 5 ways that antibiotics can affect bacterial targets: the cell wall, the protein synthesis, the cell membrane, and the nucleic acid synthesis. But the 5th one is that folic acid, which we talked about a little bit ago with the sulfa drugs and trimethoprim and that. So good job. You guys getting there. All right, what is the bacterial cell wall made out of primarily? Look at your arm. Yes, so peptidoglycan mostly. All right, Streptococcus pneumoniae, back to it, is a gram-positive bacteria. So what does that tell you about its cell envelope? So remember, the cell envelope includes everything on the outside of a cell, whether it's a wall or a membrane, anything like that. And the reason I bring that up really is because when we talk about the viruses, they can have envelopes too, but it's really only their envelopes is if they have the lipid membrane that they stole from the host cell. So they're different things when we're talking about bacterial envelopes and viral envelopes. Just reminding you guys of that. So yeah, if it's gram positive, it'll have that thick peptidoglycan layer. And something that we haven't been bringing up a lot lately is simply that if you have that thick peptidoglycan layer, it has to be supported and maintained by lipoteichoic and teichoic acid. So those always come along with it if you are gram positive. Just like how if you're gram negative, you're going to have endotoxin, aka LPS or lipopolysaccharide. Okay, Streptococcus pneumoniae is a gram-positive bacterium. So what color is it going to stain? Yeah, positive purple. Good job. The crystal violet stays stuck in there, right? Even after the alcohol decolorizer. So strep pneumoniae is known to have a capsule. What is the purpose of a capsule? Select all that apply. Okay, so capsules themselves are involved in helping to evade phagocytosis and for allowing for adherence. they're not going to necessarily be associated with endotoxin production. The reason that we can cut that out is because endotoxin is associated with a gram-negative bacteria here. And in general, just gram-negative bacteria have endotoxin. Gram-positives never do. And we know that strep pneumonia is a gram-positive. We just said it in like the last 3 slides. So, so we know that if it has a capsule, it can't be that, right? So we can cut that one out. So it's just those two, helps evade the phagocytosis, makes it harder for the phagocytes to eat them up. And then adherence, just sticking to things, the sticky coating, sugar-based. So what does the strepto portion mean in Streptococcus pneumoniae? That's exactly right. Chains of cells. You've had this shoved down your throat enough. I'm pretty sure most of you guys are getting that one. All right, what does the coccus portion of Streptococcus pneumoniae mean? All right, so that is going to be round cell shape. We're getting pretty close. Only got a few more. I say a few more, like 20 more, but still, it's a lot less than we started out with. Erica's mom calls 911 after Erica's first dose of antibiotic. What type of hypersensitivity is Erica likely having? I couldn't fit all the words in here of what's going on, but right, imagine that she's having a really hard time suddenly breathing. She can't take much air in, and yeah. Exactly. This is going to be a type 1, which is the allergy related hypersensitivity, which this is anaphylaxis, right? Which is systemic allergic response. All right, medics report that Erica was having difficulty breathing and her blood pressure was low. What kind of type 1 reaction is this? I just freaking said it. Hopefully these are the things you guys will remember. I guess technically it's 2 of these. Yeah, so technically it is allergic and anaphylaxis, but more specifically, we're going with anaphylaxis here. So, yeah. All right, what is the best treatment for anaphylaxis? You can select more than one on this one. Okay, so epinephrine and adrenaline, they're the same thing.
So why did I get it wrong?
Did you select both of them?
Yeah, but I also selected the other things we give them too, so sorry.
Okay. Sorry. All right, imagine I put my pen in my mouth and then loaned it to you, and later you got COVID. What's the pen in this? Exactly, that's a fomite. That's the term for it. So I did put my pen in my mouth. I did loan it to you, and you did get COVID. Assuming that I never had symptoms, what could I be in this situation? Right, so the correct term here is carrier. Very good, you guys. Didn't get tripped up by anything there. All right, the golden rule was DNA to RNA to protein, but which enzyme flipped that whole idea on its head? What? Yeah, so that's reverse transcriptase. You guys are doing great. Look at you learning things. All right, HIV codes for reverse transcriptase. Which of the following viruses also have reverse transcriptase? See if you guys can remember. I definitely talked about it. This is one of like the coolest tidbits. All right, so this one is just one you would have to know. This is hepatitis B. This is that one where it is a DNA virus. Then when it goes DNA, RNA, protein, it takes from that RNA and turns it back into DNA with the reverse transcriptase. And that reverse engineered DNA is what it puts into your genome. And because of that, can cause liver cancer. That's the term. All right. Transcription is the generation of mRNA from the DNA blueprint. What is the term for making protein from the mRNA copy? Yep, translation. Good job, guys. Okay, we have a 24-year-old Sonia. She is presenting with cloudy urine and pain urinating. So it's a UTI. What's the most common cause? Yeah, so that's going to be E. coli. About 80% of cases are caused by it. Probably never going to forget that either. All right, Sonia's doctor prescribes tetracycline. How does that antibiotic work? Let's see if you remember. Okay, so this tetracycline affects protein synthesis. More specifically, a lot of them that affect protein synthesis are going to affect the ability of the ribosome to either associate with mRNA or just to move along it or something like that. So the ribosome, basically. E. coli is non-motile gram-positive cocci with a capsule. True or false? Okay, so E. coli, I'll tell you exactly what's wrong with this whole statement. This is more than one thing. So E. coli, it is motile. It's one of the more like commonly motile things. And if I were to show you a sample of, that's usually what we use in the lab if I was going to show you a specific organism that was motile. E. coli is reliable for that. It is gram negative. It is a bacillus, so it's rod shaped. It does have a capsule. So if you guys remember the capsule stain slide on the lab exam, the one with the background and the halo around the clearly rods, right? That was E. coli. So. Antibiotic wreaks havoc on Sonia's biota. Now she's presenting with an uncomfortable yeast infection, also known as. All right, vaginitis is the answer here. So inflammation of the vagina is part of this. That can be caused by two organisms, the yeast Candida albicans or Trichomonas vaginalis, which is sexually transmitted, which kind of went over in the other review. So what is the hallmark feature of vaginitis as opposed to vaginosis? What's the difference? Right. So vaginal inflammation, that itis is what's giving that away. This, I don't know how helpful this will be for the guys, but I know for the ladies, they may be familiar with the fact that BV is bacterial vaginosis. So if you can remember that the bacteria are associated with vaginosis because BV, then that's osis, and itis would then have to be associated with, we know, yeast infection. That would be one of them. So Candida albicans, at least you could remember that. If not, also the Trichomonas vaginalis, which is not a bacteria either. Neither of those are, right? They're both eukaryotes, but Candida is going to be fungi and Trichomonas vaginalis is protozoa. All right, moving on. Probably all of that's the next questions. What is a common treatment for a yeast infection? Great job, guys. So that is going to be miconazole. Pretty, safe bet here with that azole added on. This is in fact what's in monostat. All right, what is the scientific name of the yeast that is associated with vaginitis? I said it a few slides ago. Let's see if you remember. Great. So this is Candida albicans. Candida, you hear people talk about Candida a lot lately, I feel like. Stupid reasons usually, I'm just saying. But all of these, just FYI, are fungi. They're all going to fall into that fungal category. Saccharomyces cerevisiae is used in like bread, like cooking and stuff like that. And coccidioides and cryptococcus are involved in meningitis, fungal meningitis. Okay, which other organism is associated with vaginitis? I did say, I knew, I should have known. See if you remember which one it is. Small. They're also brown. Yes, so Trichomonas vaginalis. What type of organism is Trichomonas vaginalis? Let's see if you remember what I said. I'm pretty sure the next question is the more specific version of it. Yeah, so this is a eukaryote, proper eukaryote. So as far as what we have talked about primarily, and definitely what you're going to be tested over, if something is not a eukaryote, if it is a prokaryote, then I would just say bacteria, because it would be bacteria or archaea. For your purposes, just think bacteria. I know we talk about the one archaea in like the dental caries. I'm not going to ask you about it, so don't worry about that. So just think bacteria on that side. If it's not a bacteria, eukaryote. Okay, yeah, yes. Okay, so this is the next part of it. What type of eukaryote is it? This is protozoan, which I did say already. So yeah, no surprise there. You guys get that correct. All right, how is it transmitted? Again, another thing I said. Yeah, that's all right. Double review, right? Yes, it is sexually transmitted infection. Okay, so how would you treat Trichomonas vaginalis? See if you can remember this one, because I feel like it can be a little trick. I didn't mean to make a joke over it. It just came out. Okay. So metronidazole is the answer here. So it is an azole, and normally we would think that that is going to be for fungal infections, but metronidazole is the deviant. So remember how some of the categories have deviants, right? So that's the problem. We have to go back and remember about those. So metronidazole is not usually used to treat fungal infections. It is used for protozoan infections primarily, as well as anaerobic infections, which will treat like Clostridium infections with metronidazole. If you have C. diff, they'll usually treat it with metronidazole. But yeah, so that one is the deviant from the azoles. So the way I remembered it, I know it's stupid. I know I've said it once and probably heard it and think it's stupid also, but I just remember it because of metronidazole. It's like metro, like it's going to the city, the metro. And the other guys aren't cool enough to go to the metro. I don't know. All right. Which of the following can be part of the normal biota? Select all of them that can be part of the normal biota of somebody. Okay, so we have Staphylococcus aureus and E. coli, which most of us got. Great job with that. You can also have Neisseria meningitis as part of your normal flora. We didn't talk about that much, but FYI, hey, surprise that you can have that as part of your normal flora. It lives usually in like your upper respiratory tracts, like in your throat and stuff like that. It's kind of creepy, not creepy, I guess, but scary to think about. Discomforting. Right now, the opposite. Which of these are only pathogens when they're growing in the human body, but are never part of the normal biota that we are aware of? All right, so yes, this is going to be most of these. So let's go through these just for a second. We know Candida albicans can be part of the normal biota, right? So it's just normal yeast. You should have it as part of your normal healthy flora, especially as a female in the genital area in small numbers, right? So it's taking the antibiotics, knocking out the normal bacterial flora that causes the yeast to overgrow and cause problems. Okay, so Bordetella pertussis, that is whooping cough. Yersinia pestis, that's the plague. Borrelia burgdorferi, that's Lyme disease. Trypanosoma cruzi, well, I mean, that's Chagas disease. And then Corynebacterium diphtheriae, that one is diphtheria. I mean, this is pretty straightforward. So yeah, so those are definitely disease-causing organisms. You would never have Yersinia pestis as part of your flora. That would not make any sense ever. So cool. I think that's all I'm going to say about that. There's a couple more. There's one more question, then maybe like a we're done thing. So catabolism, it means breaking bonds, just like how cats destroy. True or false? It's catabolism, breaking bonds. Yes, it is. Cats destroy. That's what catabolism is as well. Yep, so that is it. Okay, so that's all we're going to be doing on here for this review. I said 2 hours, but I really just meant it was probably going to go over 1 hour. I wanted you guys to be sure you had the time to sit through it. So I will post this recording online for you guys to see me say the same things over and over again. And I will post this as an assignment as well as a quiz, so you can quiz yourself with it. And I have several other Kahoots that have a lot more questions, some of which you won't be necessarily tested over, but would be, possibly useful for testing your knowledge anyways. And I'll post those as well. So do you guys have any questions for me before I go?
So the other Kahoots, are those the ones that you already have in the announcements, or will those be new ones under assignments that we can use?
Yeah, so good question. They will be new ones. I have not posted them yet because I wanted to wait for people to actually participate in this review before I just, laid out everything I had. I didn't want like one person showing up, even though it was what, like 11 in the 12, something like that. But it is what it is, right?
Yeah. And then 10 a.m. tomorrow for the Monday, Wednesday class?
Yes, Monday, Wednesday, you guys 10 a.m. in the same place as always. Not anything different. So
Okay.
if you guys are, I know most of the online people, most of them, not all of them, are going to be taking it at their normal lab time in the normal lab location. So I'll see you guys then. Tuesday, Thursday, people, morning, 10 a.m. We're going to be in 201, just like we always have our lectures. And then my evening class, 5:45 in 100. So should be same time, same place as we've been having lecture the whole time. All right. We're good? Okay, because my dogs can't take it anymore. So I'll go ahead and this will take a little bit for it to kind of upload before I can post it. But yeah, I'll work on getting the other Kahoots posted for you guys so you can start studying. You have a little while yet. And if you haven't done your the extra credit evaluations, that has to be done by midnight. Otherwise you don't get your extra credit. So knock it out.
Hey, I have a question about the extra credit. So after we complete the evaluation,
Mhm.
you just want us to take a picture of that, right? And then
Yeah, it's just the screen
upload
that
that?
says like you finished it basically. Just yeah,
Okay,
upload that. Yeah. I know it's weird, but that's all you have. So
okay.
yeah. All right, we good?
Thank you.
Thanks, guys. Thanks for showing up and good luck. All right, bye.